All right, so one of the first things that this lab is going to look at is surface area. Okay, we need to see the relationship that surface area and volume have with the rate of diffusion. Okay, so we're going to be using three different size cubes made out of a material called agar. Agar is a biological growth medium. It's what you see at the bottom of petri dishes, like a hard jello kind of deal. And they usually dissolve nutrients in it so bacteria have food to eat. We're just using plain agar, and we're mixing it with this chemical called phenolphthalein. Now let me show you quick what phenolphthalein does. Phenolphthalein is an indicator, so you can see it right here in this test tube. It's just clear. And now watch what happens if it mixes with a base. Ooh, turns pink in the presence of a base. So we are going to have phenolphthalein dissolved into the agar that we're going to have cut into three different size cubes. We're then going to soak the cubes in a basic liquid, and just as you saw there, the cubes are going to begin to turn pink. Now, to find the surface area of a cube, you'll remember it's just length times width times 6, because 6 is the number of sides that a cube has. To find the volume, it's going to be length times width times height. Okay, so that is what our two units of measurements that we're going to have for here. And then to compare them, we want to see the surface area to volume ratio. So you'll have to measure the surface area to volume ratio as well. So we're going to soak them for a set amount of time, probably about 10 minutes, 10 to 15 minutes. And then what's going to happen is really cool. We're going to cut these cubes in half. All right, so we have a perfect square. And what you're going to see is that basic solution is going to dissolve at the same rate from all sides. So what you're going to see, as long as you don't let it soak all the way through, you'll see a perfect cube that the basic solution hasn't touched, and everything that it has touched is going to be a bright pink color, because that's showing diffusion. So we're going to compare how much of the cube has been touched by the basic substance. I want to see the percentage of the cube touched by the substance in the large cube, the medium cube, and the small cube. And I want to see if the size of the cube has an effect on the rate of diffusion in the cube, and which one is more efficient at undergoing diffusion. All right, so that's the first part of the lab. If you need help with the calculations, I'll help you on lab day. All right, the next part of the lab uses dialysis tubing to model the cell membrane. I'm going to show you how to make a bag in a second, but essentially it's going to look something like this. You're going to tie both ends like an open water balloon. Like if a water balloon were open at both sides, you have to tie it at both sides. That's what it would look like. You're going to have a series of solutions. There's going to be salt water, sucrose water, glucose water. I'm going to have some egg whites also that you could use to put in there. And you're going to soak them in any of those solutions too. All right, so you're going to have a solution inside the bag and another solution outside of the bag. And you're going to see which way water is going to move through that membrane, or if you think things other than water are moving through that membrane. But dialysis tubing is kind of set up like this. It's got pores, all right, that are very small. So some substances are going to be able to move through there very easily. All right, they could fit nice and easily through these pores. Other larger molecules aren't going to be able to fit through those pores. All right, so this models a cell membrane because that makes this selectively permeable, just like a cell membrane is. All right, so let's take a look at an example of that. I'm going to show you an example of the dialysis tubing in action and show how it's a semi-permeable membrane. So I have some materials here. I have a solution here with glucose and starch mixed in it. I have a starch indicator, iodine, which tests for starches. So I'll show you how that works also. So here I have some of my starch and glucose solution. Now, when iodine comes in contact with a starch, you're going to see a color change. All right, it turns like a very dark purple, almost like a black, because of the starch in there. So when starch touches iodine, that's what we get. All right, and I also have these testing strips. They're for urinalysis. They could tell you if there's a buildup of glucose in your urine. All right, we're not going to use them for urine today. We're just going to use them to test glucose. So let's just see. Here's what a test strip looks like. There's the color of it. This is before it's been tested. So now I'm going to put it into our solution of glucose and starch, so we should get a positive test. So I'm just going to dip it in to the solution, remove it, and let it sit for a couple minutes, and we should see a color change. All right, so let me show you the dialysis tubing bag. It's kind of a pain to work with. I've done it hundreds of times, so I have some techniques that may help. There we go, that's better. Nice long piece of tubing. So here's the tubing. It's like a piece of tape, but it's actually a tube, so it can be hollow, but it's really, really thin. All right, so we need to open it. So what I like to do is I like to take the top half, or like any half, fold it, and just kind of rub it between your fingers, like that, and that will allow it to open. So now I have an open side. I got it going a little bit, and I'm going to tie it just like a water balloon. So I'm going to twist it. All right, make a little loop like so, and force the open end through that. All right, so now I have a knot at one end, so it's closed. At the other end, I'm going to do the same thing. I need to open it. All right, so rub it down. Now I have some separation between the two halves, and I'll be able to fill it up. So now I'm going to take my starch and glucose solution. I'm going to use the pipette, and I'm just going to throw some into the bag. All right, so we have some of that solution in the bag. I'm just going to tie this off now. Same way. And tie it off. Now you're going to have all those different solutions I mentioned before, either sucrose, glucose. We could try starch if you want starch, albumin, salt, and see which way the water will diffuse. So if that's what you're doing, you're going to take the mass of this before you soak it, and then take the mass after, so you can see which way the water has moved. So let me just show you real quick. We have a positive test for glucose in there. You can see it turn brown. Now I have an environment that we're going to soak it in. This is just distilled water, so there's nothing in there. And I'm just going to put a couple drops of iodine into the water. All right. All right. Mix that around. So it should be a nice pale yellow color. All right. So now we have that. I have our bag, and we're going to let this soak in here for a while, and I'll come back and show you what that looks like in a minute. Alright, so about 20 minutes has gone by, so let's see what has happened with our dialysis tubing. So here is the system. You can see that the water and the iodine is still the same color, but we have a color change inside of the bag. Alright, so now if you remember from before, what makes starch turn this color is iodine. Okay, so iodine must have gotten in the bag. However, it did not get into the environment. Alright, because if it did, the environment would be that color also. So that means that the membrane of the dialysis tubing is semi-permeable. All right, I'm just going to test this for glucose now. So I'm going to take a glucose strip. It's clean. Give it a little dip in here. Let it wait a second. We'll see if we get a change in there. Now beforehand, there was no glucose in the environment. It was just water and iodine, so we would have got a negative test. Now I'm going to see if it got through the membrane and it went from inside of the bag to outside of the bag, because glucose is much smaller than starch. Glucose is a monosaccharide. Starch is a polysaccharide. So starch is going to be a lot bigger. So those holes that we saw in the dialysis tubing might not be able to get through as easily. But we did see that the iodine is able to get through the bag, but the starch was not able to get through the bag. And I'm already starting to see a color change with the test strip. So if we look there, it's already starting to get a little more brown. And if I wait a couple more minutes, it'll be completely brown. So maybe I'll put a little screenshot of that in there, but I don't want to keep up your time. I know it's very precious. So there's an example dialysis tubing showing how it's semi-permeable. Water was able to get through, iodine was able to get through, starch was not able to get through, so it hit a wall. Okay, and we'll see glucose in a second. All right, the next part of the lab is observing plasmolysis and how osmosis works in plant cells. So I'm going to go to Petco. I'm going to get some aquarium plants, and we're going to put some salt water on those and look at them under a microscope. That's pretty self-explanatory. We'll do that tomorrow. But the last part has to do with the potatoes that we've been talking about in class. You guys have been designing your own experiment to figure out which concentration are all of these unknown liquids. So I'm going to have a red, a blue, a green, a yellow, and a purple liquid. All right, I'm going to label them A, B, C, D, and E. Now there's going to be a water, a 0.2, a 0.4, a 0.6, and a 0.8 molar sucrose solution in each of those, but you're not going to know which one. And you're going to have to use potato to figure out which solution is which. So hopefully you guys have already figured out a way that you're going to do that. We've talked about it. You've showed me a couple things. You guys have had some good ideas. So what we're going to do is take the potato, maybe like 5 pieces per container, and you're going to have to measure them before they soak somehow. So choose a way that you think that would best show how to measure the rate of osmosis. And you're going to soak them. All right, so we'll put them in A. And then we'll soak 3 and put them in B. And C and D and E, etc. All right? So you could, some of you guys are measuring them every 5 minutes and seeing if there's a change. Some of you want to leave them overnight. Some of you want to wait 15 minutes and measure them. So whatever you guys think, we'll see. We'll compare our results and we'll see what we get. But you're going to have to figure out, is water, I think water is C, B, I think A is 0.8, D is 4, E is 6. All right, I'm just making these up. I don't know what I'm going to do yet. I haven't made the solutions yet. But I need you to figure out which one is which based on the rate of osmosis in these potatoes. Okay? And I will give you bonus points if you could figure out a way to tell me the molarity of the potatoes. All right, so that's all the explanation I'm giving you for that, because you guys have been working on that by yourselves. So if you have any questions, shoot me an email. Or if you're John, you can just tweet me like you do every other night. All right, so I'll see you guys in lab.